Welcome back, everybody, to the third session in today's Successes in Neuroscience Symposium. Those of you who have been with us from the beginning, or even from partway through, will agree with me, I think, that the presentations have been really, really interesting. Many similar issues have been raised among researchers coming from quite different directions. So it's been remarkable, I think, and we have more. So 3 more speakers, and the first one is Dr. Gerlinde Metz. She's a professor of neuroscience and a Tier 1 Board of Governors Research Chair at the Canadian Centre for Behavioral Neuroscience at the University of Lethbridge. Her team is looking to build resilience in people, especially those most vulnerable to tragedies like natural disaster or war. How do you do that? Part of this research explores the influence of experience and environment on behavior and brain plasticity. Dr. Metz.
Hello, everyone, and thank you very much for your kind introduction. And first of all, I'd like to thank the organizers for inviting me here to showcase some of the research that's going on at the University of Lethbridge. So with Valentine's Day coming up soon, first slide, please. Thank you, Craig. I would like to talk a little bit about our brain on love and relationships. So what happens when we miss those vital ingredients to our lives? And exactly this has become a very central issue for our society during the ongoing COVID pandemic right now. So next slide, please, Craig. Before the COVID pandemic hit us so hard, would you have thought about the impact of social isolation on our physical and mental health? I guess right now the news shows to us every day on a daily basis the unfortunate circumstance of the pandemic and how it affects our social lives so very much. So it was actually a coincidence that our lab, our team in Lethbridge, has been investigating the effects of social isolation on health for a few years before the pandemic came on board and joined us. And so it turns out that actually the most vulnerable members of our society are also the most affected by social isolation. And those are elderly, children, and pregnant mothers. Next slide, please. So under normal circumstances, about 80% of mothers would report that they experience low to moderate or even severe distress at some point during their pregnancy. Next, please. And during the COVID pandemic, this rate has increased drastically. Now we have at least twice the rates of anxiety and depression that we see usually. Next, please. So one reason for this extra stress is that we're missing out on physical connectedness. On one hand, the physical distancing is absolutely necessary to fight the pandemic and combat the virus and restrict it. But on the other hand, it also makes many of us feeling more lonely, and we're missing out on critical family and community support. And this actually generates a paradox, which is very difficult to solve. In addition, the loneliness and missing social support may also fuel worries about finances and job security, home office work, the closing of day homes, kindergarten, schools, and universities. Plus, the situation has also led to a dramatic increase in domestic violence and fewer options to seek support. So in our research, we've tried to capture at least some of these aspects. Next slide, please. So some of our work in rats and animal models of social isolation done by Mirella Ambeskovich and Jane Sabo. This work has revealed the effects of social isolation, and I show you some of the data here. You see the single housing condition SH in the blue bar and the paired housing, social housing condition shown in red in those bar graphs. And social isolation elevated the stress response and reduced the motivation and motor control in a feeding task. So the animals that were socially isolated needed longer to respond to a reward and eat a Cheerio. We see the parallel here with also Dr. Gibbs' work that she was showing in children. So we love those Cheerio experiments. So on the left-hand side, you can see the social isolation and eating the Cheerio. On the right-hand side, we have a condition where those socially isolated animals were in addition exposed to an experimental mild stressor, and they became more frantic and actually became faster eaters and more motivated to frantically reach for those Cheerios. So the latency measurements then were not different from the controls anymore, but we wanted to tease out those differences even further. So in addition, therefore, next slide please, Jamshid Faraji and my team has shown that social isolation limits the ability of stress coping along with changes in the hippocampus, such as reduced hippocampal volume. And rats exposed to social isolation, those are groups indicated by an SI. It's kind of small if you have a small screen, I apologize for that. But I just want to bring you across the general message that there's lower expression of brain-derived neurotrophic factor, BDNF, in the hippocampus in animals that experience social isolation. And this is very important because it limits neuronal plasticity and ultimately impairs the ability of the hippocampus to help regulate the stress response. Next slide, please. So what is also important is that social isolation induces a biological fingerprint, a very personal fingerprint for every individual. There can be something like a methylation mark attached to the DNA, or the expression of a small non-coding RNA molecule, such as a microRNA, or a downstream metabolic signature. And when socially isolated rats get pregnant, they may actually pass on some of these marks to their offspring. Next slide, please. So as we know, some of these epigenetic markers are potentially heritable, and they can be passed on to the next generation. And next slide, please. And even to the grand offspring, and even further generations. So this is like a fast-forward process of evolution, and it helps us to rapidly adapt to a dynamically changing environment. So it's very important that we have this mechanism. Next slide, please. Biologically, these heritable epigenetic signatures so help us to adapt to, for example, to a challenging environment if we are imprinted or programmed by stress. And this helps us to thrive and survive in a stressful environment. Next, please. The issue is that this adaptation also comes at an expense, and this might be a higher risk of disease later on in life. And the intergenerational and transgenerational consequences of the COVID pandemic and all that's going on, including the social isolation, still remains to be seen in future generations to come. Next slide, please. What we know so far is that social isolation and any other major stressor can lead to very robust patterns of epigenetically inherited markers. And this is what we can see here in Stephanie King's recent work on microRNA signatures propagating across 3 generations of rats in cortex and placenta. So shown here are different variations of lineages whose ancestors experience stress. And you see those generations, F1, F2, F3. And even on the small screen, you probably notice this very robust inheritance of microRNA signatures across the different generations. So some of these microRNAs are very stable and they just don't disappear after a few generations. They're going to stay and change the brain plasticity levels, change behavior, and make us the people and personalities that we are. So it is important to point out that in our experiments also we found that placenta often reflects what we find in the cortex, as you can see here, the red bars and the blue bars. And we assume, therefore, that the placenta offers a very useful tissue or proxy target to predict how the brain will develop. Next slide, please. So in addition, Keiko McCreary's work on our team has shown that in addition to these microRNA signatures, F3 offspring born to a lineage of socially isolated mothers are generally more stress sensitive. The first blue bar here indicates non-stress controls, and the second and third blue bar indicates animals born to a lineage whose great-grandmothers were exposed to stress. And while this is in corticosterone levels at the basal resting state, these data are not significant. You see a trend. And when you look at behavior and certain stressful situations, this difference between stressed and non-stressed lineage will really open up to be significant. So animals' behavior, for example, metabolism and epigenetics, actually will show much stronger effects to ancestral stress. And this is remarkable because remember, we are only talking about great-grandmothers being stressed. So the stressed offspring will be more like the competitive, impatient, restless type A personalities in the animal world. Next slide, please. Fortunately, we are able to do something about this. We can expose these animals to social enrichment, a chance to play with many friends. And you see the red bars here illustrate that rats exposed to social enrichment become more laid-back, happy hippie rats across the board in any of those lineages. To illustrate to you a bit more of the changes we see in behavior, I'd like to show you an example of behavioral traits. So in the next video clip, next slide, please, you see the water competition task. I hope that's working for us. Yes. These rats are thirsty and they're competing over access to drinking water. And what we see is that socially isolated rats tend to be more aggressive, but as soon as they're housed with friends, they become friendlier and more cooperative and willing to share resources. So you've seen the differences here. The non-marked white rat at the bottom was a bit more competitive in this case. So next, we actually have this, so thank you. The mechanism behind this involves oxytocin, which is a small peptide hormone with really big functions. And we can also call oxytocin the cuddle hormone because it works like the candlelight dinner equivalent of the stress response. So Jamshid Faraji in my team has discovered that oxytocin is producing the beneficial actions of social support that we see, especially in females. Rats housed socially have higher oxytocin levels, and you can see that on the left. And here on the right, you can also see that social housing in rats elongates telomeres, a marker for biological aging. And telomeres are like the protective caps sitting at the end of a chromosome, very much like a shoelace. This is my sneaker, and you see the shoelace ends. They're covered by plastic tips, and so they're preventing the plastic tips, or the telomeres in the case of the chromosome, will prevent those ends of the chromosomes from fraying and becoming fuzzy. So they're protective caps. This is as close as my research ever gets to footwear technology. So with every cell division, the telomeres get shorter, and longer telomeres indicate a younger biological age. And stress would lead to shorter telomeres, and social support would result in longer telomeres. This means that social support slows down biological aging and promotes a longer lifespan, particularly in females again. So if we block the effects of oxytocin, this is again something that Jamshid Faraji has shown, we also block the benefit of social support, and this we can translate to humans. So to learn something about the impact of social isolation on our health. Next slide, please. One outstanding example of prolonged social isolation is someone going on a space mission. So we have been setting up a partnership with NASA and received urine and blood samples from astronauts who went to spend some time on the International Space Station for extended periods of time in some cases. Michael Gaaynggal, in collaboration with Tony Montina from the Department of Chemistry and Biochemistry here in Lethbridge, have found that a space mission drastically perturbs the human metabolome. We saw clear metabolic effects of entry into space and return to Earth. The time points here depicted as blue or pink blobs. And these are males on the left-hand side and females on the right-hand side at different time points. You get the idea, even with this is small. It's just to show you an illustration of how metabolomic data look like as a result. And the overall message here is that males actually adapt much faster to life in space than females. And an issue is, of course, with the space travel, we also have confounding effects of radiation and zero gravity and a kind of other stressors in our way. So if we want to talk about social isolation in a particularly isolated way, we need to come up with another experiment. So to control for these effects, we also worked with a different project, the Mars 500 mission. Next slide, please, Craig. So the Mars 500 study is a psychosocial experiment conducted by Roscosmos in Russia, the European Space Agency in China, with the goal to prepare for future human spaceflight to planet Mars. And within this experiment, the crews lived and worked in a mock-up spacecraft for 520 days and actually stayed on planet Earth. So they did not leave into space. And these are very fresh data that I'm showing you here. But what you see is really a time series showing that the longer the isolation lasted, the more separated became the metabolic changes, the pink and blue blobs that you see here, and they were drifting apart over time. So how can we use this knowledge to promote health? Well, we don't want to stay in gloom and doomy states here. Next slide, please. One example of how we're using this work is the work we do with vulnerable human populations, such as the Australian Indigenous population, in a collaboration with Kim Ray. Dr. Ray is using social and arts therapy to overcome trauma in the Gomeroi Gaaynggal people, who as children were forcibly removed from their families, very much like residential school in our Canadian First Nations population. So this therapy that Dr. Ray applies is showing an amazing impact in healing their wounds of the past. And we observed that it also heals some of the metabolic disturbances. So next slide, please. In addition to this work, Josh Heightened, with the help of Tony Montina from chemistry, discovered metabolic markers that predict who's most vulnerable to stress and in need of such a therapy. So for example, Josh is working with people affected by the 2013 Calgary flood. And from these findings, we're learning how to identify those who are most sensitive to social isolation and stress and who are more resilient. And we also will get to know this way, for this route, who is most in need of interventions and mitigation therapies to reduce potential health risks. So next slide, please. So my take-home message, maybe also with Valentine's Day, keeping in mind, is that social support can reduce stress and build resilience, and probably also help us stay healthy through the COVID pandemic. And if these days hugs are not possible, sometimes even a virtual meeting like this can do a pretty good job, I think. So next slide, please. The data that I've shown are a huge group effort, and I would like to thank my most amazing team of trainees and collaborators who have made this research possible. And I also would like to thank Campus Alberta for facilitating our many networking and social interactions that lead to research such as the one I've shown you. So next slide, please. I would like to conclude with some words by Taiki Masura from Harvard University. Despite all of the talk about resilience, being an individual trait, most of us are only as resilient as we are loved. And next slide, please. With this being said, thank you so much for tuning in and for joining us today.
Thank you very much, Dr. Metz. I do have some questions. The work with NASA is really interesting to me because I think of isolation as having almost no human contact. And, you know, there are lots of people who would probably think that having another 5 people around is as much contact as they might like. So do you see what I'm getting at? Like, you know, what really defines, because you see metabolic effects in even a handful of people on the space station. So, you know, what is the perfect situation? Do you need contacts with a dozen or 24 people?
So this is a very good question. Thank you, Dr. Ingram, for raising this point. There's been quite a few studies, of course, on social isolation, and often they consider having at least one friend or one physical or one face-to-face interaction per week would be a pretty good achievement for someone who's older and maybe living in a senior's home or being in an isolated rural area. But on the other hand, I think it also depends very much on the mindset. And when we're really living in a space station, I can imagine those small crews, 5, 6 people at a time, maybe, they do have virtual connections also with their families, and they have a lot of contact with planet Earth, but they're still isolated and in a confined environment for a longer period of time. So you probably end up seeing a mixture of feeling lonely in some ways, missing family, missing friends for that extended period of time, but also being constrained physically in the smaller space. So it's hard to dissociate it. that in some ways. And it's actually really difficult to find good human study populations to address these questions in some more detail.
Right. So, given what you've shown about the transgenerational effect of isolation, that would suggest that identifying people who might be suffering from social isolation is best done as early as possible, not just for the sake of the patient, but for the fact that if they get past childbearing age, then you've exposed new individuals to the same sort of effect. Is that right?
Yeah, I think this is a really important question because especially in human populations, we haven't considered those inter- and transgenerational consequences enough yet. So we actually have created a tool that we can hand out to populations of our studies to address the question about the ancestors. This is not usually asked in a study. How did your mom feel and your dad? What are their experiences? Have your ancestors been through a war? Have they been forcibly replaced or displaced from the environment where they're forced to migrate? And so these are important questions. And what we're studying now as a result of this, we're studying collaborations with populations around the world to address imminent questions like this, because as we have climate change moving on, we have more populations afflicted not only by natural disasters, which we are very interested in, as you mentioned, but also by other consequences that come along with climate change. And I'm very fortunate to be part of really fruitful, productive collaborations there within Alberta and around the world to address these questions.
Well, thanks again, Dr. Metz, and you will join us again for the roundtable after the next two speakers. Thanks.